Hello, friends. Welcome to part 2 of 1.3 membrane structure. All right, in our last notes, we talked about how the membrane is made of the lipids and the cholesterols, the three main components. Well, there it is, right? So we're talking about how the structure of the membrane thing that goes around our cells and keeps everything protected, right? Regulates what goes in and out. And so last time we were talking about the phospholipids and the cholesterol, and this time separate lecture on the proteins of the cell membrane. You can see right here, show you guys some drawings later of things that you have to identify and how we'll have you draw those, just like the parts of the cells, right? But as you can see here from the cell membrane, let me see, why is this not? Oh, sorry. There are different types of proteins here, right? You've got the globular proteins. You have a protein channel, right, that goes from in the cell to out the cell, right? You've got these peripheral proteins, the integral proteins, the surface proteins. So proteins do a lot with our cell membranes, and we'll talk about the different functions of them. So why we have the proteins, and then a little bit about their positions, and then how to draw them. Okay. All right. So let's start with what we use the proteins for. So here are 4 different examples of proteins. One for transporting, receptors, enzymes, and anchors. We talked a little bit about anchors before, right? Things that keep them on. But you do need to know that proteins have many different functions within the cell membrane, right? So if you had to look at like the 6 functions of proteins in the cell membrane, you would have to come up with those from memory, be able to draw them, right? To be able to identify them. So those are the 3 kind of like difficult things that we will learn to do. All right. All right. So the first one, and then on the next one, I have, just so you know, a mnemonic device here for you, Jet Rat, but we're going to go over and I'm going to explain these a little bit more. So if you want to pause it, write down the functions and then unpause and add some extra things. Okay. All right. So first of all, proteins are responsible for hormone binding sites, right? So hormones are fats that go around in our body. You probably know a little bit about hormones, right? So you've got growth hormones, you've got estrogen, testosterone, you've got things that for your digestive system, right? So all those different hormones in our body. But if you ever thought about, because they're transported in the blood, right, so if it's in the blood and it's going around everywhere, why do only certain cells react to those hormones? So you can think about like the growth stimulating hormone or follicle stimulating hormone for hair, right? And so there's a certain part, especially, moving this around, right, during puberty when more hormones are released, right? So we see, like, especially in males, like the production of facial hair, right? So you've got like specific places where you get facial hair growing, but that stimulating hormone to grow hair is in the blood everywhere. But there are specialized cells that have these receptors for the hormones that send that signal. Okay, we're going to start stimulating follicle here to grow here, right? So that's why we have those certain patterns of hair growth, right? Whether it's on our head, places in our body, or like our face, because there's certain cells that have that receptor for the hormone. If it doesn't have that receptor, it just keeps going by, right? So that's one of the things that we use, or that proteins for in the membranes are for. Okay. Another thing is for enzymes. So proteins are enzymes. That's hormone binding site. So immobilized enzymes with the active site on the outside, right? So we're looking for, so here, inside, outside, right? So we're looking for enzymes here, active site, and it's on the outside. And that means like it's immobilized. So, right. So it's not moving. So some enzymes are moving around our blood, right? Whether it's ducts, things like that in our body, and some of them stay there. And the active site where that substrate attaches to is on the outside, and that'll create some kind of signal for the cell, or some type of reaction. So enzymes are proteins, specialized proteins. We'll have a whole separate section on enzymes, and you guys should get excited about that. But for now, just know that they are part of the cell membrane. Okay. Another function of those proteins is adhesion to form tight junctions between cells. So they actually form as like little like interlocking, right, in between cells, so they don't move around. So there's certain proteins that connect so that they adhesion to cells around them. And then cell to cell communication. So there's a couple different examples we just went over, but proteins on the outside are how one cell is going to communicate with another cell, right? They're the little receptors for those messages. You can think of like that. Okay. Another way that proteins are embedded in our membranes are for transport. So we don't want everything going in and out of the cell, right? We want to be able to regulate things that are let in and let out. If you have, I know some of you are in AP Psych and you've already talked about the sodium potassium pump, right? So that there's sodium and potassium that go through the outside of that neuron. That is a transport protein, right? So I get transport proteins that open up. They let certain things in, they let in certain things out. So they're kind of like the gatekeeper, right? The bouncer of the cell. And then the other things are for transport. So you can either have passive or you can have active. Passive transport does not require energy. Remember that. And then active transport does require energy. So it needs that molecule of ATP as well. And we have not talked about ATP, but I know some of you guys are asking about the mitochondria and production of ATP. It's a molecule of power, right? So on energy, all the power, adenosine triphosphate. Okay. All right. Here's a mnemonic device to help us remember those 6 things. You can think of the jet rat, right? The jet rat. So again, going through all of those different things that it did, but gives it a little bit of a mnemonic device to help you remember. So junctions, keeping the cells together. Enzymes, right? So attached an enzyme for localized activity, transport, passive, active, in and out, to recognize certain cells, right, for those hormones, the anchor or attachment sites, and then transduction for those hormones, right? Transduction means sending that message, okay? Jet rat. They do, like, remember those 6 different things. If you can literally just remember those JETRAT, then that will help you remember, like, okay, junction, right? You can try and remember what those things stand for instead of just blindly memorizing those 6 functions. All right, second part, we've got 2 different groups of proteins. I will show you in your book too, but let's make sure that we have this down. So you've got integral and then peripheral, and use your knowledge of your other words and, your context of other words and other situations of what it means to be integrated into something and to be on the periphery, right? So if you've gone through driver's ed, you know that the periphery is on the outside, right? It's outside your central vision. Integral means to be connected or through, right? To be completely integrated means to be all the way through. All right, so the integrated proteins going there, these are permanently attached. They do not move, right? So they are part of the cell membrane. They have, going back to when we were talking about hydrophilic and phobic, the phobic, right, side is at least one part of their surface, right? So they have that part of the surface that does not like the water, so it points that way. And that part is embedded in the hydrocarbon tails and the phospholipid, right? Because they are also hydrophobic. So you can think that they kind of pair up there, okay? Some are what we call transmembrane, so across, trans meaning across, right? So that means they go all the way from one side to the other, in to out, right? Other ones are just on one side, but they're still stuck there, right? So they span across the membrane if they're transmembrane, and the hydrophilic part's projecting all the way to the outside. So instead of just sitting there in the little, right, this is the electron microscope, so sitting here with the tails, right, you actually get them going all the way through right here. All right, I'll show you what this looks like in your book in a second. All right, the peripheral proteins, you can think are the opposite. So their attachment is reversal or temporary. So it can just go in for a short amount of time and then keep moving on, or it's reversible, right? So it can be kicked out. The hydrophilic on their surface, so it's not embedded in the membrane, right? It doesn't go all the way through to the hydrophobic tails there. And they're usually found attached to the integral proteins. So I get those integral proteins that have those, I think it was on the last one, I had the anchor sites, and they attach the peripheral proteins, so they work together there. Some have a hydrochain carbon, I'm going to show you that in a second, that inserts into the membrane and anchors the protein, so it has like a little chain that kind of goes in. You can think of like a hook that attaches itself into the membrane, and some do not. Some, go on and they attach to this protein. Okay, so integral and peripheral, need to know those 2 types of hormone categories. Let me hold on just a second. Okay, so I'm going to show you your book here in a second. I will open that up. But a lot of variety, right, in what proteins do in our bodies, and it's totally dependent on the cell, right? So I need certain cells in certain parts of my bodies that have receptors for, like, glucose, for insulin, like things like that, for different types of hormones, and then some that might not need anything, that are just structure cells. They keep like things, the structure together, or they're just dividing, and those membranes don't have a lot. Okay, so the protein content determines the function of the cell. So don't know what type of cell, if you know what type of cell it is, then you should be able to predict the proteins that are in those membranes. The more active membranes have more proteins, and the less active ones have less, right? And you can imagine that if I don't have any proteins in the membrane to let things in and out, or to receive messages, or to have anchors, then that cell's not going to be doing much except for like keeping the structure. Okay. All right. So how we're going to draw those? So in your book, I know I opened this up, but I opened a ton of stuff up. Let's see, here it is. Okay, so when we're drawing membrane structures, this is on page 31 in your book. We are going to, just like in the last one, know how to do, just like the organelles, like how we want to draw it out, right? So here is the diagram that's like a simplified version of the one I showed you at the beginning of class that shows you those hydrophilic and hydrophobic, the phosphate groups in the heads right here, right? The tails are together. This is showing, so those are our phospholipids. It's showing the integral proteins, right? Going from one side to another. And then the peripheral proteins these are the ones in purple, right? So here's an integrated one that doesn't go all the way through. Here's a transmembrane one that does go all the way through. And then the cholesterols, right? So the cholesterols here that have those little chains that go off. I'm sorry. And then I've got some saturated fats and some unsaturated fats here as well. All right. So some examples of when we draw those, and when I'm just going to put these in your notes so you can go back and look while you are actually drawing. Let's see, here it is. Okay. So when you are drawing those things, right, it might look something very simple like this, right? So I've got the cytoplasm. So I'm showing the inside of the cell and then the outside of the cell. It shows an integrated protein right here, right? And then I've got my carbon chain right here, my sugar chain here, my glycoprotein. I've got my phospholipid bilayer, but then I also have the phospholipid. Here you go. There we go. Shown separately, and then it has the philic and the phobic tail. Okay, so it's got integral protein, peripheral protein, inside of the cell, outside of the cell, the entire bilayer, and then the separate parts of it, the phospholipid with the hydro, this and that. Okay, here's another example, right? Super simple. We're not trying to complicate anything. I've got that carbon chain here. I've got the transmembrane protein. I've got, this thing Miss Myers is blocking, the carbohydrate, the glycoprotein, the phospholipid. So we'll practice drawing all these things out, but know that you should be able to name all those and then label all those too, right? So if you see a picture of it. Even this, I liked this one too because it's very simple. I draw this, about that, and then I have everything to the side, and you can even memorize those integral, head, tail, cholesterol, glycoproteins, and then peripheral proteins, but not like this. Oh, so messy. Look at this. Just make sure we're taking our time when we draw things and having them correctly labeled. See how this, so why is this wrong? So it's like, it's not necessarily even the handwriting. I can read the handwriting, but look, right? The glycoprotein not completely attached to what it's pointing to, right? Integral protein that has that extra little, remember that doodad that goes that way, we don't want that. The peripheral protein is not even attached to the cell membrane, it's completely peripheral, right? And then the protein channel I can see here, and that one's not too bad, neither is this phospholipid bilayer. But some of these, we lose all these points because they're not either not attached, the too much complete circles, or it's not pointing exactly where it's supposed to go. Okay, I'm not going to even talk about this one. You know who you are. No, I'm kidding. This is from last year. All right, so that is, there we go. Okay, we're going to go back over that in class. So we are all finished with our part 2, the proteins. Lots of information there, but again, using those mnemonic devices and what we know of the words integral and peripheral and the jet rat should help us jog our memory when we have to recall these things. Okay, I will see you in class and have a fabulous day.